Hey everyone, thanks for taking the time to listen to this recording on the part of the class we missed today. Just want to keep you up to speed on the cell cycle. We're going to get through this a little bit. We're going to talk about tumor suppressor genes, proto-oncogenes, and we're going to stop there and then we'll deal with mitosis itself next week. But really where we're at right now is discussing the cell cycle, how the cell cycle goes through its stages and begins the process of replicating the cell. So the thing with the cell cycle is it's broken down into different stages. There's a growth stage, function stage, replication stage, and it's really a set of repeating events. And so the first thing we're going to deal with is growth. So growth is going to be your G1, G2, and there is even growth in S, which is when DNA replication occurs. Function itself is G1 or G0. So if you notice, for growth and function, it can be both G1. So you can have a functioning cell in G1, or you can have it growing in G1. It depends on what you want to do with the cell. And then you have replication itself. That's the M phase. That's the mitotic phase. That's when it's actually going to split. We're going to go through that as well. So if we look at the cell cycle itself, we can see here it looks like a circle, and it's good to idealize and diagram it as a circle, because with that idea, this cycle is going to be repetitive. So it begins in one part, moves through the cycle, and begins over again. And where it's going to begin and end is going to be in G1. So G1 itself, when it's in that phase, that is function. So when it's inside of G1, you could have a cell. It could stay in that stage in G1 or G0 for the next 40 years if it wants to, or it could stay in G1 for about 8 hours. It really depends on what you want to do with that cell as far as does it need to replicate or does it not need to. So think, for example, about a neuronal cell. A neuronal cell in the brain may never replicate once you're an adult and you've gone through initial growth. So that may stay in G1 or G0 for 80 years, depending upon your lifespan. Or if you're a liver cell, it might only stay there for about 8 hours, or especially like a skin cell, if you've got a cut and needs to replicate, it might only be there for about 8 hours because it's moving through things. So in general, we call G1, G2, we call these interphase. And so that's when it's going to be doing normal functioning, not in the mitotic phase, and it's moving through that sort of phase in itself. We can see there the G1 resting stage or function stage. The S phase comes after G1. That's when the DNA is going to synthesize. So we're going to double, or we call it DNA synthesis. That's when we're going to double our DNA in S phase. G2 is when everything else is going to replicate. So, if we're going to go through mitosis and create two cells, the mitochondria you have there, also the microtubule spindles you need to form, they're going to develop inside of G2 because the rest of the cell is now getting ready. So, and then finally you go into M phase, which you see here at the top, and that's broken into 5 different stages: prophase, prometaphase, metaphase, anaphase, and telophase. And then the whole thing ends with cytokinesis, which is the pulling apart of the cell. So that's different cytokinesis than mitosis itself. We put them all together like we usually call it PMAT plus C. I'll show you that on Monday how that works. But in reality, if we go back and we look again, G1, this is function or growth. The cell begins to grow. It literally gets bigger. It has the energy it needs to undergo that replication. If everything looks good, it pushes into S phase, doubles its DNA. And then after that, we go into G2. Everything else begins to divide as it needs to. You're going to get the mitochondria, if you're a plant cell, chloroplast double as well. And you're going to get these microtubule spindles, which we're going to talk about on Monday. Those are going to happen as well. So then the question becomes, how long does this take? It really depends on the cell itself. Fruit fly embryos will go through the cell cycle, that entire circle, in about 8 minutes. The mature cells themselves take longer to grow. So a mammalian cell, for example, takes 24 hours to go through this process. That's the minimum, 24 hours inside of a mammalian cell like animal cells. So if you want to double or replicate a cell that you already have, make 2 daughter cells, looking at 24 hours. As you can see, growth itself, the G1, G2, and S, that takes the most amount of time. That takes about 23 hours. But we can see here M phase only takes about an hour. That's all it takes. Most variation itself is going to be the length of G1. Like I said, it can stay there for years if it wants to. It can stay there for days, or it can spend about 8 hours in that cycle. Now, even when we talk about liver cells and how we double or grow, liver cells can grow faster if they need to. I think this is an interesting picture to look at. There's something called a hepatectomy. With that, the idea is with hepatectomy, you're taking part of the liver, you're going to resect it out, and you can give it as a donation to somebody else. So liver donation is a real thing. Individuals can do it. They need to plan ahead for it because you need to have a really, really healthy liver in good condition to do this process. But the idea is if you give a partial liver to somebody else, and even your own liver, it will regenerate on its own. And you can see this process, in fact, under those conditions happens very quickly. Within usually about 30 to 90 days, The liver, even if you resect part of it, remove part of the liver, you regenerate and have complete mass recovery. That's in a healthy liver. You don't see that in livers that have fatty acid or fatty liver disease. Fatty liver disease can happen because of two main reasons. One can be alcohol related. So if somebody consumes too much alcohol, they can get fatty liver disease. And you can see it's a fatty liver. If you do a resection, you have mass recovery failure. It doesn't become as full size again. So it could be because of drinking too much alcohol. If somebody stops drinking alcohol, fatty liver disease ends or takes about 2 weeks to recover from, and the liver can restore itself. So the liver is a very, very good organ, vital organ that can do self-recovery if we give it the right conditions to do so. If it's not alcohol related, it's probably related to excess weight, obesity, and in which case, to have fatty liver disease reverse itself, the person needs to lose weight first. Now, how do we tell a cell to actually undergo mitosis, undergo splitting or division into two daughter cells? It's going to happen because of growth factors. So growth factors, as you can see, you have something that's intracellular. It's sitting on the external environment of the cell and also in the internal environment of the cell. Some growth factor hits it, and when it hits it, it tells it and gives a signal to say, this is a signal that you need to grow. So external to the cell, there's been some stimulus released. It can be, going back to the example, imagine that you've got a cut on your skin. You've lost skin cells. Growth factors will be released in that region to tell the epithelial cells, double. We need to fill in the gap where we lost skin cells. And so that growth factor hits from the outside and creates a cascade-like event inside the cell. You can see there's different proteins here called the RAS and the RAF and the MEK and the ERKs, etc. They're all going to move through the cytoplasm, eventually make their way into the nucleus, and interact with different growth factors there, as well as tumor suppressor genes that tell the cell it's time to split, it's time to divide and grow. So that's going to be the process. So you're going to use these growth factors external to tell the cell to grow. In addition to that, you're going to use something called checkpoints. So with the checkpoints themselves, these are established to make sure that mitosis proceeds correctly and the cell is ready for division. There are three main checkpoints inside of the cell cycle. One happens in G1, one happens at the end of G2, and one happens in the M phase. We can see with the G1 phase, the checkpoint there is going to be more than anything else to check the nutritional state of the cell and what the size of the cell is and whether or not it's gotten a growth factor. So let's say a cell tells itself, I need to divide. The cell in this G1S checkpoint is going to be saying to itself, have I gotten a growth factor signal to divide? If it has, it says, yes, that's good. At the same time, the cell is going to check its nutritional state. Does it have enough ATP? Does it have enough reserves to actually undergo the cell cycle division process? If it does, it's also going to pass that. And finally, has it increased its overall size? Because again, it's going to split into two daughter cells. You don't want them to be half the size of the original cell. You want them to be about 3/4, or maybe 7/8 the size of the original cell. So it has to grow itself in size before that. If it reaches all those points, it passes the checkpoint and tells the cell, okay, we can leave G1, move into S phase, double our DNA, go into G2, begin to double the mitochondria, and then you're going to hit one more checkpoint at the end of G2. You can see it's the G2M checkpoint, so it's watching that gate, and more than anything else, what it's doing in that stage is making sure that DNA synthesis has occurred. It's making sure all the DNA has been doubled and that the DNA is intact. In other words, there's no major damage to it. There hasn't been a lot of mutations. So it's checking for DNA integrity at the same time. If that all looks good, it proceeds immediately into M phase. This is going to be the mitotic phase. This is when you're splitting apart. At the spindle checkpoint, something called metaphase, we're going to talk about that on Monday. That's when they're going to get one last checkpoint. If everything is good, it says, good, let's go, let's divide, boom, pushes through that, goes through cytokinesis, and the cells are back in G1. And now at this point, when you come back into G1, you've got two daughter cells, and everything should have worked perfectly. So these checkpoints are there to make sure the cell is ready. If the cell's not ready, it halts cell cycle division. So imagine you get to the end of G1. If you're not getting a growth factor signal, the cell does not move forward to divide. If everything checks out there, but it gets to the end of the G2 phase and it notices things are wrong with its DNA, it also will not divide. It might instead decide to do apoptosis, or in other words, cell destruction in a formed way to actually not hurt the cells around it. So these checkpoints are vital. The checkpoints are there to make sure the cell actually knows it should be dividing, and when it's dividing, it's making two clones, two daughter cells that are the same as the first cell. If it can't do that, it can lead to problems in the cell. And one of those problems, of course, is cancer. So what is cancer? Cancer is unrestrained, uncontrolled growth of cells. And really what it is a failure of that cell cycle control. When we go back and look here and we see the cell cycle and how it cycles through, what we're seeing is we lose control of that in this phase. What can cause this? What can cause a disturbance of the cell cycle where the cell doesn't know that it shouldn't be undergoing that process? It can be caused by either a problem or a mutation in tumor suppressor genes, or a mutation in something called proto-oncogenes, which is another protein your body can make. So we're going to go through these very briefly. Here's the idea with tumor suppressor genes. So this is a very classic protein, by the way. It's called P53. When we talk about cancer nowadays and what happens with people, everybody's going to talk about P53. If you are dealing with an oncologist at any point in your life, they're going to be looking at P53 expression levels or even checking the integrity of P53 in your cells to see whether or not it's mutated, depending upon where the cancer is located. Here's how it works. If we look at the top image here with P53, normal P53, what it does is it detects DNA damage. So if your DNA has been damaged by heat, radiation, some sort of chemical shift, it detects that damage, and what it does is it halts cell division. It says, I am not a healthy cell right now. I have DNA damage, and to think about actually dividing myself before I do that or continue to maintain and function as a cell, I need to repair that DNA damage. So the P53 protein activates. It's going to kick in DNA repair enzymes. It's going to stop that first checkpoint. It's going to halt DNA synthesis. And if it repairs itself, if it can repair its DNA damage, as you can see on the top part here, it says everything is good. P53 then turns back off, allows the cell to divide, and everything is good because you've got a normal healthy cell. Let's say instead, looking at the top pathway here, you can't repair the DNA damage. In that case, instead, P53 triggers apoptosis, which is controlled destruction of the cell, making sure all the DNA is destroyed as well. It gets rid of that cell. It says this cell is no longer good. We're going to get rid of it. It's no good for you. That's if you have a normally functioning P53. Let's say, however, you have a mutation in the P53 protein itself. Something went wrong and it damaged the P53 protein. In that case, when you get DNA damage, it can't be recognized by the P53 to halt DNA synthesis and cell replication. In that case, the cell begins to divide itself without repairing its DNA. So now you have a cell that's divided itself. One side, of course, is going to get normal healthy DNA because you're diploid. Those cells are going to continue to divide normally. But on the bottom, you can start to accumulate more and more mutations. If P53 is inactivated, it's not stopping, for example, when damage happens in proto-oncogenes or damage happens in other genes. So you get this cascade of events where you get more and more mutations in your DNA. It can affect more and more genes that are important for cell cycle control. And if you hit enough of those genes, the tumor suppressor genes like P53 and also proto-oncogenes, now the cell goes into uncontrolled growth. And when that happens, as we can see through the process, eventually you can create cancer cells. So what does this mean? First of all, it means just because you have an abnormal P53 or you have damage to your DNA, that doesn't mean you're going to develop cancer. There's a lot of other mutations that have to happen within the cell to reach the stage of cancerous cell growth, where it's uncontrolled, unrestrained cell growth. But it means if you do have an abnormal P53 or an abnormal tumor suppressor gene that's been mutated and that hasn't been repaired, it's more likely that you may develop cancer in the future. And that's why we look out for that. We try to look for these, like for example, we do gene sequencing nowadays. We look for P53 to see if it's abnormal or mutated, because again, that's an indication about cancer that might develop in the future. Here's an example about how this plays out. Besides the P53 gene itself, there's another gene called RB. RB stands for retinoblastoma gene. And so this gene is responsible for a disease called retinoblastoma. You can see the image on the upper right-hand side. This is an infant. Usually retinoblastoma impacts people 8 years or younger. This is a very young child. This child is probably like a year old, maybe 2 years at max. And we can see here, somebody took a picture of them with a flash, but they're only one eye. One eye looks more like the pupil is black. One eye, the pupil looks like what we call a cat's reflex. So in fact, this is called a cat's reflex reaction. When we see the whitening in only one eye versus both eyes in an image, it means to us that possibly there's a tumorous growth on the retina itself. And that's what this image is actually seeing. The flash hit, and it's seeing the tumorous growth on the back of that retina. If you took a picture of this of a child and it shows like this, it's a visit to the eye doctor almost immediately. You want to go take them to the physician to begin with. They'll do a check. If everything looks fine, it might be the end of it. Or they might say, we're gonna refer you to an ophthalmologist to take another look. Because again, if it's retinoblastoma, if treated early, it can be cured. It's a disease that can be handled nowadays. If it goes too long, it might result in nucleation of the eye, which means removal of the eye itself. And it might be metastatic, which means it spreads to the body and the individual can die. So early detection is always important. What happens in this disease is a tumor suppressor gene as well. So you got P53, you got RB. They're both tumor suppressor genes. In this case, with this protein, the same thing. It's not stopping cell cycle. It's not giving that checkpoint for DNA integrity not being intact. Usually, retinoblastoma happens because it's already in the familial line. It's generational, so it's passed from generation to generation. If an individual has one bad copy of RB, because remember we're diploid, if they have one bad copy, that means they've still got one good copy. But if that other copy gets mutated, that's when you have the problem. And that's what happens with people with retinoblastoma, where they had usually one good copy, and then just by happen chance, the other copy gets damaged, and then that leads to cancer later on down the line. So again, paying attention to these things, knowing about tumor suppressor genes, their role in stopping cell cycle and arresting it if there's DNA damage, extremely important. The flip side of this is the proto-oncogenes, and as I talked about, these are the genes that are going to tell your cell we're receiving a signal to grow and divide. So inside the cell, you have these different oncogenes, and with these oncogenes, the idea is they are supposed to get an external signal, something outside the cell as a growth factor telling them to grow. But imagine one of these genes gets a mutation in it, and it thinks it's on. So in other words, like the idea with proteins, as we're going to show you, and I'll even show you on the next page, maybe it's even easier to see it. You can see here you've got that RAS protein and SRC kinase. These genes, these proteins are turned on by a growth factor, but imagine they're just always on. Imagine they had a mutation which makes them think, I received the signal. So imagine RAS gets mutated. If RAS gets mutated, it's going to think it's on. It's then going to tell SRC, let's move forward, and it's going to cause that cascade event to tell the cell, I'm getting an external signal to tell me to grow. In reality, there is no signal. That's the problem with these proto-oncogenes. They've turned themselves on and told the cell, I'm getting a signal to grow when no signal was given. That's your problem with the proto-oncogenes. So the proto-oncogenes are saying, grow, 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 grow. Your tumor suppressor genes are saying, stop, 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 if DNA isn't intact. So when both of those things go wrong, when you've got a proto-oncogene which has been mutated and also your tumor suppressor gene is mutated, you've lost cell cycle control because it's being told all the time to grow and there's nothing inside the cells telling it, stop, something's actually damaged with my DNA, and if I can't fix it, then I need to cause apoptosis or take myself out. Look at the possibility or probability of these things being involved in cancer. The RAS protein mutation activated in 20-30% of all cancers. Look at the P53 protein, mutated in 50% of all cancers. So it doesn't mean every cancer is going to have exactly these tumor suppressor genes or these proto-oncogenes mutated, but they are big players. So again, that's why we look at these nowadays. If you know anybody that has been diagnosed with cancer in the future, you do, they will be looking and talking about this. So nowadays, because of the ability we have with gene sequencing and getting that information on an individual basis, they will take samples, they will look for expression cycles or expression signals of these different genes to see if they're on, to see if they're being activated, to see if they've been mutated, all these sort of things. Because again, it's going to tell the oncologist and the medical staff what's going on at the molecular level. And that might be why we're seeing cancer developing the way it is. Here's a video for you to watch. I like to show you these videos themselves. Really helps you to show a little bit more about the cell cycle. I do recommend you watch it. You're going to get this also in the homework. And in reality, that brings us up to mitosis. And that's where we're going to stop this lecture. I wanted to keep it short, but I wanted to give you a little bit of a background on cancer, on the cell cycle itself. When we come back together on Monday, we're going to go through mitosis. We're also going to have some time on Monday to go through and look at, do an in-class example. So I'll send that in some instructions to you also through email as far as how to know what to do with that. So that's it, folks, for today. I wish you all a very pleasant weekend. We'll see you on Monday.